Okay, for lecture number 8, we're going to move on to our final steps of digestion, where we're going to completely break apart these organic nutrients that we've taken in from our foods, things like proteins and carbohydrates and lipids and so forth. And we're going to chop them into their monomers, which are the forms that can actually be absorbed across the epithelial lining of the small intestine. All right, so as I mentioned earlier, the chyme that's coming in from the stomach, the organic nutrients in our foods have not been completely chopped apart into their individual building blocks or individual monomers. So they're partially digested at this point, your carbohydrates and your proteins. Your fats are mostly undigested. Yes, you do have some lipases that are produced by the tongue and the stomach, but most of the fats for the most part are not digested. Once the chyme enters the small intestine, it's going to spend about 3 to 6 hours in the small intestine. Most water gets absorbed in the small intestine, and almost all of these nutrients will wind up being absorbed. And go back and review if you don't know this terminology yet, you should. The building blocks of carbohydrates are monosaccharides or sugars. The building blocks of proteins are amino acids. The building blocks of most of the lipids we're discussing here are glycerol and fatty acids. Those building blocks are what get absorbed across the epithelial lining of the small intestine. So the job of these enzymes in the small intestine is to completely chop apart these larger molecules into those building blocks, and that's what we're going to be concerned about here. All right, so let's talk in a little bit more detail about digestion of carbohydrates. Carbohydrates are chains of sugars, but you've got to chop those chains into the individual sugars or monosaccharides before they can be absorbed across the intestinal lining. Okay, and the main ones are glucose, fructose, and galactose. We hear about glucose all the time. Fructose is a sugar that's part of sucrose, table sugar. It's also present in fruit. Sometimes it's called fruit sugar. Galactose you'll find in a lot of dairy products. It's part of the lactose that's present in dairy products. Okay, so you have a series of enzymes that gradually break apart your carbohydrates until you get to the point where you have these individual sugar. So these include the salivary amylase that we've talked about before. Those are enzymes that chop apart polysaccharides. And pancreatic amylase is produced by the pancreas, does a similar type of thing. And then along the brush border, remember the cells that make up the epithelial lining of the small intestine, they have that brush border or microvilli along the top, and you have lots of enzymes embedded within the membrane there. Those are called brush border enzymes, and their job is to finish off the processes of chopping apart these chains into individual building blocks or molecules. So those include enzymes with names like dextrinase, glucoamylase, lactase, maltase, and sucrase. And no, we're not going to talk about what all of those do. They're just keep in mind that there are, enzymes are very specific about what they chop apart. So you need lots of different enzymes to chop apart very specific molecules as part of your digestion. Let's take a look at a couple of these though. Sucrose is table sugar. That is a glucose and a fructose linked together. So every time you eat table sugar, that's what you're swallowing. And along the brush border, you have an enzyme called sucrase, and what sucrase does is it chops apart glucose and fructose. So now they're separated from each other. And those two sugars can be absorbed into the epithelial cells that line the small intestine. Sucrose cannot. So you've got to be able to chop apart that sucrose. Lactose is glucose bonded to galactose sugars. So that's a disaccharide. You've got two sugars linked together. Lactose is present, as you guys know, in a lot of dairy products. All right, so along the brush border, you have an enzyme called lactase. And what lactase does is it chops apart the glucose and the galactose. So you now have those simple sugars, those monosaccharides, and those can be absorbed into the cells that line the intestinal tract. People who are lactose intolerant either lack these lactase enzymes, they don't have them in their brush border, or maybe they don't have as many of them, and because of that, they can't chop apart glucose and galactose very easily. So the lactose molecules, those disaccharides, stay within the lumen of the small intestine. They're not actually able to absorb them. And so that can cause intestinal irritation. And another thing that it does, if you think about it, lactose is a solute. Solutes attract water. So if you have all this undigested lactose passing through your intestinal tract, those lactose molecules are attracting water out of the cells that line the intestinal tract. So it will increase the water content of the material passing through your intestinal tract. And I'll let your imagination run wild. With that, you can figure out what's going to happen then if you are lactose intolerant. Okay, so some more about digestion of carbohydrates. Starches, long chains of sugars, again, they get partially chopped apart by salivary amylase, pancreatic amylase. The salivary amylase doesn't completely chop apart these polysaccharides. Instead, if you think of what it's really doing, if you think about a polysaccharide being a long chain of sugars, and sometimes these chains actually branch and become pretty elaborate. The amylases, what they do is they chop apart these big polysaccharide chains into smaller pieces. Okay, and then your brush border enzymes, things like dextrinase and glucoamylase and lactase and maltase, etc., they're going to act on these smaller pieces, those are called oligosaccharides, and we'll chop those into the individual monomers or individual sugars. And again, the brush border enzymes are very specific. You know, as I was just showing you, lactase only breaks apart lactose. Sucrase only breaks apart sucrose. Maltase only breaks apart maltose. Maltose molecules are two glucoses that are linked together. So you need lots of enzymes that perform very specific functions. You know, I do want to, just a brief timeout. A lot of times students, and I think it's because they hear the most about enzymes and A&P when you're covering digestion, and so a lot of students get in their minds that enzymes only break things down, and that's not true. Yes, with digestion, we're talking about enzymes that break bigger molecules into smaller molecules, but you also have building enzymes, enzymes that take smaller building blocks and create the larger molecules that our cells need to function. So keep in mind you need enzymes for almost any kind of chemical reaction that's taking place inside the body, both building and breaking things down. We're just right now focusing in on the ones that break things down. All right, the flow chart here from your textbook really is just kind of providing an overview in a graphical form of what I was just talking about. Now, the last step that has to take place is absorption once you have those simple sugars. And if you think about the cells that are lining the inside of the small intestine, you've got that brush border or microvilli up here. And you have learned before, if this is a cell membrane like you would have up here on the brush border, hydrophilic solutes like sugars cannot freely pass across a cell membrane. Instead, they've got to have some sort of membrane protein which allows them to pass through. Okay, so these products of digestion, these monomers, these building blocks that we're learning about, they're going to be transported through membrane proteins into these epithelial cells that line the small intestine. And then what happens is they pass across these cells, and they're released out on the other end of the cell, which is also called the basal end of the cell. And from there, they're going to enter the connective tissues that underlie the cells there, and they're going to wind up being taken in by capillaries in the intestinal wall. Okay, boom, they're going to move into the capillaries, and once they're in the capillaries, those capillaries are going to lead to the veins that drain the small intestine, and those veins are going to lead to either the, let's see, most of the veins that drain the small intestine lead to the superior mesenteric vein, which connects to your hepatic portal vein, which then carries this blood to the liver, because the liver then is going to process a lot of those nutrients that we're talking about. All right, how about digestion of proteins? Okay, the building blocks of proteins are amino acids. All right, as we've already talked about, in the stomach you do have an enzyme called pepsin. Go back and review about how the gastric glands actually produce pepsinogen, the inactive form that gets converted to pepsin at a low pH. And also the low pH of the stomach, if this is a folded up protein with a 3D structure, the low pH denatures the protein. And then these pepsin enzymes can come along and chop that denatured unfolded protein into shorter chains of amino acids. And that's what's entering into the duodenum. You have partially digested proteins. Those partially digested proteins then encounter the trypsin, the chymotrypsin, and the carboxypeptidase enzymes that are part of your pancreatic secretions that have become activated in the duodenum, like we talked about in an earlier lecture. Those are going to continue the processes of chopping apart those proteins. And then finally, you have brush border enzymes that are going to take these short chains of amino acids and convert them into individual amino acids. And then these are absorbable. Those are going to be absorbed across the brush border and into the cells that line the intestinal tract. Some terminology with proteins. Polypeptide is a name for a fairly long chain of amino acids. Peptide is a shorter chain of amino acids, but just learn that those three terms, protein, polypeptide, and peptide, all three are referring to chains of amino acids. Peptides are shorter, polypeptides are longer, and a polypeptide that is folded up into a 3D shape is referred to as a protein. And then peptides get broken apart into amino acids, as we've been talking about repeatedly. Okay, this diagram from your textbook is showing you what's going on. All right, so these are the cells that line the, these are the cells that line the lumen of the small intestine, the epithelial cells. And they're showing you just like what we talked about with the carbohydrates. All right, so these are your short chains of amino acids, your almost completely digested proteins. And then you have pancreatic proteases and brush border enzymes that finish off chopping those into individual amino acids. And then right here, what they're showing you is that those amino acids move across membrane proteins that transport them inside the cells. That occurs through active transport. If you go back and review that information from Biology 201 or an earlier biology course, active transport is a process by which solutes are pumped from one side of a membrane to the other. It actually takes energy to make that happen, and that's because they're moving against the way they want to naturally move. They're actually more concentrated in here than out here, so you've got to pump them from outside to inside to force that to happen. And then the amino acids travel to the basal end of the cell, and you have membrane proteins there that transport those across, and then they move into the capillaries, like we were talking about with the carbohydrates. And then the capillaries they'll flow through the blood. The amino acids will be carried to the liver through the hepatic portal circulation. All right, and this is just a table from your textbook, which is providing an overview of what we've already been discussing. All right, how about your lipids? Lipids are not quite as easy to break apart because lipids are hydrophobic. So they're not freely dissolved in water. And yep, that brings up another point, terminology. Dissolve versus digest. Dissolve means, like, if a molecule is dissolved in water, that does not mean it is broken apart in the water. It just means you've got your solute here, like glucose, for example, and it is surrounded by water molecules. Or like with, well, let's think about sugar crystals, for example. When you see table sugar, sucrose, sucrose is, so you got sucrose. All right, sucrose crystals are where you have a whole bunch of sucrose molecules forming a chunk. When we say they're dissolved in a fluid, that means you have now individual sucroses that are surrounded by water. That's what dissolving means. That does not mean the sucroses have been broken apart or digested. Not at all. Digestion of sucrose again is a glucose and a fructose bonded together. When you break that apart into glucose plus fructose, you have digested the sucrose. So don't confuse digestion and dissolve. That does not mean the same thing. All right, same thing with fats. Lipids do not dissolve in water. They don't become surrounded by the water in our body fluids very easily because they are hydrophobic. And that's where those bile salts came in, like we talked about in an earlier lecture. The bile salts produced by the liver, and which are contained in the bile, surround fat molecules and help them become covered with water around the outside. So they help break apart big fat globules, and that's called emulsification. But notice what it's telling you here. Bile does not break the bonds between the monomers. So it's not digesting them, it's just helping to dissolve them or emulsify them in our body fluids. We need lipase enzymes to break apart lipids. The tongue makes some, lingual lipases, the stomach makes some. The pancreatic lipases are going to do the bulk of the job. And the goal here, most lipids that we take in from our diet are triglycerides, like when you eat a fatty steak or something, all the fat in the steak, most of that consists of triglyceride type lipids. And the building blocks of those are glycerol and fatty acids. Here's a pretty good diagram from your textbook, which is showing you, here's a big blob of fat coming in from your foods that you've been eating. Here are the bile salts that got squirted into the duodenum as part of your bile. And notice they are coating the fats because the bile salts have a hydrophobic portion over here and a hydrophilic portion over there. The hydrophilic portions face water. The hydrophobic portions over here are facing the fats from the fat globule. So they're able to coat and make those fat globules or droplets much, much, much smaller. When they do that, those are called micelles, by the way, where the bile salts have surrounded little tiny droplets of fats. And then your lipase enzymes can break apart those fats, those triglycerides, into glycerol and fatty acids. Well, and really I need to clarify because, let's see, let me erase this. Really the enzymes take your triglycerides and convert them into a monoglyceride plus 2 fatty acids. A triglyceride is a glycerol. There's glycerol, and it's bonded to 3 fatty acids. Okay, so what the enzymes that digest your fats really do is they chop off 2 of those fatty acids, and that leaves you with 1 fatty acid connected to a glycerol. That is called a monoglyceride. Mono because it has 1 fatty acid attached. So it's really your fatty acids and these monoglycerides are what get absorbed across the intestinal lining. All right, so inside the intestinal lining cells, all right, so fatty molecules, these fatty acids and these monoglycerides that have come from your foods that you've digested, they don't dissolve in the watery-based fluids of your cells well because they're still fatty. So we need something that's going to package them up and help hide them from the watery-based fluids of your cells so that they can be transported more easily. And so those little particles that these fatty acids and monoglycerides are packaged up inside are called chylomicrons. So they're little particles that consist of proteins that protect and kind of hide the fats from the water with all these fatty acids and monoglycerides on the inside. And those little particles get transported across the intestinal lining and they get picked up by the lacteals. So if you guys remember, the lacteals are special lymphatic vessels that you have along the small intestine, and those villi, those hills that we have along the inside lining of the small intestine, whose job it is to pick up these chylomicrons, these little particles of fats that are coming in from the diet. So fats actually get carried away from the intestine in the lymph first, rather than the blood. And this table from your textbook is just reviewing what we talked about, the step-by-step breakdown and absorption of fats from the diet. And they do mention here there are some fatty acids, some short-chain fatty acids. Fatty acids are mostly carbons and hydrogens in long chains linked together. Some are shorter, some are longer. Different triglycerides will have different types of fatty acids in them. And so some of the short ones do actually get absorbed into the blood, like some of the or the other nutrients that we're discussing. So that's what's being mentioned there on the table. All right, finally, our 4th type of major organic macromolecule, your nucleic acids, DNA and RNA. All right, so since we generally eat foods that consist of plant cells and animal cells, those are formerly living things. You still have DNA and RNA. Those are nucleic acids in there. So we need enzymes to break those molecules down as well. So the pancreas makes ribonuclease that digests RNA molecules, and deoxyribonuclease breaks apart DNA molecules. So it breaks them apart into their nucleotide monomers. Nucleotides consist of three parts. All right, you've got a nitrogen containing base, and that's the part that is designated as an A, a G, a C, a T, or a U if you're thinking about RNA. And you have a sugar, which is a pentose sugar, which means it has 5 carbons in it. And then you've got phosphate. Those are the three parts of a nucleotide. So you've got enzymes that split those three things apart as well. So that's what they're talking about there with the brush border, enzymes. So nucleotides are the building blocks of DNA and RNA, and then those nucleotides themselves have three major parts that get chopped apart prior to absorption. Okay, and then there's a little diagram from your textbook, which is showing what we just talked about. All right, again, a little bit more about absorption. All of your nutrients get absorbed, or essentially all of your nutrients get absorbed in the small intestine. And as we've seen, most of the digestion occurs in the small intestine as well. So that's there's a misconception. Most people think, well, my food gets digested in my stomach. No, most digestion takes place in your small intestine. Some people think they're absorbing nutrients in their stomach. No, almost all nutrients are absorbed in the small intestine. And most of this absorption takes place prior to the ileum. So remember, your small intestine has the duodenum, which is actually fairly short. Next is the ileum, and then finally you have the jejunum. You have the duodenum, the jejunum, and then the ileum is the third section, ileum with an E. So most of this absorption happens before you even get to the ileum section of the small intestine. The ileum, though, is the part that really reclaims the bile salts. Remember how we talked about the bile salts get recycled, reused, they get sent back through the bloodstream to the liver, and that tells the liver it's time to make more bile. You also absorb electrolytes, things like sodium and calcium and chloride and potassium in your food and beverages. Those have to be absorbed as well across the epithelial lining of the small intestine. And water as well. The water from your food and beverages, most of that gets absorbed across the cells that line the small intestine also. Most nutrients get absorbed by active transport. And again, there's a cell lining the small intestine. You have proteins up in the brush border, not the enzymes, but proteins whose job it is to transport things like sugars or amino acids or electrolytes from the lumen of the small intestine inside those cells, and then they get transferred, most of them, into capillaries to be transported to various parts of the body. But just remember, most of the fats wind up in those little chylomicron particles that get taken up by the lacteal lymphatic vessels within the wall of the small intestine. Okay, an active transport, again, is where you move a solute across the cell membrane by pumping it, forcing it through a membrane protein, a channel or a carrier, to allow that to happen. All right, interestingly, how does water get absorbed? We have lots of water absorption going on throughout the day. 9 liters, now that doesn't necessarily mean in one day you're drinking 9 liters of fluids, but your GI tract makes lots and lots of secretions, bile, pancreatic juice, etc., the chyme from the stomach. So there's a lot of volume in there as well, and you need to absorb the water back out of the lumen of the small intestine and return it to the bloodstream so that you can hang on to those fluids. And most of that occurs by osmosis. So if you think about an individual cell lining the lumen of the small intestine, you've got lots of solutes in here, very high solute concentration, because it's taken up, all these nutrients are being pumped inside those cells. So water follows solutes, right? Remember, water is attracted to solutes. Water follows salt. Water follows other types of hydrophilic solutes as well. So simply because you have a much higher concentration of solutes inside the epithelial lining of the small intestine, that's going to suck water across and into those cells. So osmosis is responsible for about 95% of our water uptake. The rest of it's going to be absorbed in the large intestine, as we'll see when we talk about the colon. All right, if you have a patient or know somebody with malabsorption of nutrients, that's simply going to be anything that's interfering with these processes of absorbing nutrients. And the causes, anything that interferes with delivery of bile, like if you have a gallstone, for example, or you have some other blockage of a bile duct, or liver dysfunction, the liver's not producing enough bile. If that happens, you're not going to be able to emulsify your fats and absorb your fats. Fats are a major source of calories, so that's going to be one possible trigger of malabsorption. Anything that's interfering with pancreatic juice production or secretion, like what if you have pancreatic cancer, or even like a benign tumor of the pancreas, you're not going to, that might be blocking the pancreatic duct. You're not going to be able to release as many digestive enzymes into the duodenum. If you can't chop apart these big organic molecules into smaller pieces, you're not going to be able to absorb them. People who are gluten sensitive, you know, we're hearing a lot about that these days. Gluten is a wheat protein. And in some people, quite a number of people actually, the immune system sees that as a foreign antigen that it needs to attack. And it's really inappropriate. It's not like a molecule of a bacterium or a virus that we need to get rid of. It's seeing this wheat protein called gluten as something foreign that the immune system needs to respond to. The immune system over-responds. That's a type of hypersensitivity or allergy, and that causes inflammation along the intestinal lining, which can be very painful, and it can cause problems with absorption. If you have a lot of inflammation along the intestinal lining, absorption is not going to function properly. If the intestinal mucosa is damaged for any reason, such as through bacterial infection, there are some antibiotics that you take in pill form that might cause intestinal lining damage if you take them for a long time. Any of those types of things can interfere with absorption as well. All right, so that was a pretty long video lecture, and the next one should not be quite as long. We finally made it to the colon, the last stretch of the alimentary canal or digestive tract, and so lecture number 9 we will discuss the colon. And then the last couple of lectures for this unit, I'm actually going to spend a little bit of time talking more about how the body processes lipids, because we hear about a lot of that terminology quite a bit, and it plays a really major role in healthcare and personal health. So the last couple of lectures we're going to return to lipids and talk about them a little bit more thoroughly.